Okay, so let's wrap up talking about audition by focusing on two other key aspects of the auditory system or hardware. The reason I kind of use this vague language is that because sound localization, while it obviously has something to do with audition and auditory processing, this other domain, balance, which is involved in the inner ear, actually doesn't really map on very well to audition. So that's why I use that kind of terminology. So let's start off talking about sound localization and how your brain is able to do it. And to get an understanding of why this is an important thing to talk about, why it's kind of a computationally tricky thing, let's go back to talking about the organization of early visual cortex and early auditory cortex. Okay, so remember, in early visual cortex, it's retinotopy, in early auditory cortex, it's tonotopy. So the question is then, with a tonotopic organization, how could an auditory system tell the location of a sound source? So if your auditory system is primarily just trying to focus on frequency initially, okay, how is it going to be able to tell where that frequency is coming from? In vision, this is easy, okay? In vision, there's a spatial map. If you want to know where something came from, V1 basically can tell you very easily exactly where it is. If you light up this cell or this cell or this cell or whatever, V1 just has that nice and easy. In vision, this is easy, but there are unfortunately no spatial maps in auditory cortex, so how is it going to do it, okay? There's going to be three different main principles that we're going to talk about to answer this question about how it does it. So the first question, or the first principle, are called interaural differences, okay? So what this basically means is that if you have a sound source that is directly to your right, it actually turns out that the distance to the ear on your right side is obviously closer than the distance to the ear on your left side. And so what actually will end up happening is that the speed or the time or the moment in which that sound will get processed by the right ear will be a little bit sooner than it will get processed by the left ear, okay? So there's an actual kind of relationship here between where the source is coming from out in its location in space and your two ears. Conversely, if the sound now is a little bit like, further forward to the right, there'll still be it'll hit the right ear faster than the left ear, but at a different time. Because now, here it's hypothetically 0.3 milliseconds, is over here it's 0.6 milliseconds. So that's a cue right there, all right? So interaural differences are a main principle. Now, how are interaural differences actually processed by the brain, okay? What's going on neurologically that can allow your brain to compute these different types of things, okay? It turns out that to process interaural differences, there are these things called coincidence detectors. Now, coincidence detectors can sometimes be a tad confusing for people, I've noticed. So I've took this clip from a video because I think it describes it very well. I put the clip down here in case you want to watch the full one, because I know sometimes it can be a little trippy. So let's watch this and then we'll go over it.
Humans and other animals have the amazing ability to locate the source of a sound in three-dimensional space. A sound originating from the left side of your head will reach your left ear before it will reach your right ear. And this time difference is analyzed by neurons in the brain to extract information on sound location. In this model system, sound signals from the left ear are transmitted via the blue axons, and sound signals from the right ear are transmitted via the red axons. These axons come together to synapse onto postsynaptic neurons, here numbered 1 through 7. We assume that it takes the same amount of time for sound from the left ear to reach X as it takes for sound from the right ear to reach Y, and that these neurons, 1 through 7, are maximally activated when sound signals reach them simultaneously. In other words, these neurons act as coincidence detectors. The different lengths of these blue and red branches, called delay lines, will cause these neurons to be maximally activated by sounds at different locations on the horizontal plane. If a sound originates from straight ahead, it will reach the left ear and the right ear at exactly the same time, and therefore the sound signal will reach X and Y at exactly the same time. Only neuron 4 will be maximally activated. If a sound originates 30 degrees from the left, it will reach X slightly before Y, and only neuron 5 will be maximally activated.
Okay, so these sort of coincidence detectors are thought to be the neural implementation of interaural differences. It's a system that I think is a little tricky to explain just with my words. I found that this little animation, I think, does a little bit of a better job. So hopefully that gives you a clear sense of it. But just to kind of talk about it in my own language, just to go over it one more time, the way that you can think about it is like so. You've got these different sound sources that are color coded here based on the, the left side being blue, the right side being red, and so forth. So if a sound comes from the right side, it'll hit the right ear first, it'll get processed, it'll be going down this stream, and the source of, or like the signal won't make its way all the way down here until this blue one starts firing itself. So basically what'll happen is the sound signal comes into the right ear, it's getting processed down here, and while it's getting processed, the sound hasn't yet hit the left ear. And so basically, while this is making its way down the right ear, it's going to take a little while before it can get down to the left ear, and it won't converge on this little coincidence detector for a little while. That was you can see now why I showed you the video. That was not my most eloquent moment right there. So hopefully what you can just kind of get from this little diagram is that these coincidence detectors just correspond to these different locations in space, and that this is the neural implementation of the interaural differences. This actually was interestingly discovered and understood best in the barn owl. Barn owls use sound localization a lot to hunt, and this is the model organism. And so sometimes if you read books, you'll see people talk about coincidence detectors and these slight asymmetries in the barn owl system, okay? Now, this system alone won't do it, okay? This system alone of just interaural differences is not going to be enough to tell you where a sound is in space. Why? The reason is that because you can have these ambiguous situations. So think about this. Imagine if you have the sound directly in front of you or a sound directly behind you. In that case, the sound will hit the left and the right ear at the exact same time, but you yourself will be able to hear the difference pretty well. I mean, if you just want to try it out, snap in front of your face, snap behind your face or behind your head, and you can hear the differences, okay? And it's not just about forward and backwards. Consider this little frog. If the frog is, I don't know, 30 degrees to the left, or, 30 degrees off of, immediately to the left, then in that case, the sound will hit the left and the right ear at these respective times. If it's negative 30 degrees off of the left, the frog, the left and the right ears will get the sound from the exact same time. So again, the interaural differences wouldn't be able to tell these two apart. But your auditory system is clearly capable of doing that. If you snap like this, snap like that, you can hear the difference going on around you. So how does your brain do it? So one way it does it is with these things that are called intensity differences, okay? And it actually turns out that your head, literally your head, creates a sound shadow. And so if a sound is coming from forward and to the left of you, the intensity with which it hits the left ear will be greater than the intensity with which it hits the right ear, because of the fact that your right ear is in the sound shadow and your left ear is not. So to go back to this frog, again, the sound shadows coming from this angle or the sound shadows coming from this angle will help you understand if that frog is, in this case, coming from forward and to the left, okay? Another way that this works is that there are pressure differences between the ears. So as you'll remember, sounds are these waves. And so it actually turns out that when a signal is coming immediately from the right side of space, it'll push the right ear in, but it will actually end up pulling the left eardrum out a little bit, ever so slightly, ever so slightly. Whereas when it comes from the front, now both eardrums are going to get pushed in. And so now these pressure differences are another type of signal that allows your auditory system to have a sense of where sounds are. Interaural differences, intensity differences, pressure differences, and so forth. Before we move on real quick, what I want to actually just show you is how these sort of pressure differences and intensity differences can be used in other animals. So here, for example, is
It listens for the tiny sounds of its prey moving about below, but must take great care not to scare them away.
So notice what its head's doing as it's hunting for the little rat mole thing under the snow. Can't see it, but he can still sense it. Watch this, this is adorable. And it works. This is actually gonna work. Watch this. He comes out with a little rat mole thingamajiggy that he's gonna eat, okay? So in that case, how did it work? Why was he rotating his head? He was rotating his head because he was basically getting a bunch of different samples of the little sounds of what was coming on from the the little rodent thingy underneath the snow. By kind of moving his head, getting different pressure differences, different intraoral differences, different intensity differences, he was able to better localize where that little thing was, even though he couldn't see it. This is one of the reasons, actually, by the way, dogs, sometimes when you say kind of weird things, they'll kind of like tilt their head when they're a little bit confused, because what they're actually also doing is trying to get different samples in that way, by kind of just trying to be like, wait, I'm trying to make sense of it, so maybe if I listen this way versus listen that way. And that's why also their ears can do some of those, fun floppy things. Okay, so sound localization is thought to be from these intraoral differences, coincidence detectors. These alone won't do it, so then you can also rely on pressure differences and intensity differences. I just realized I think these are in the opposite order of the way that we went into them. Okay, so now let's wrap up this by talking about balance, and specifically talking about the role that the inner ear plays when it comes to maintaining your sense of balance. So to understand how we maintain balance, what we're going to do is we're going to go back into the inner ear, but we're going to talk now about another function of the inner ear besides converting sound waves into neural impulses, okay? And the part that we're focusing on in the inner ear is not the cochlea, but it is connected to the cochlea somewhat tangentially. And it is these little things here, these little semicircular canals here that we're going to focus on, because they're very important for maintaining balance. So what I'm going to do now, because it's kind of hard to get a sense of this entire structure from this angle, is I'm just going to show you this whole structure, the cochlea and these little semicircular canals, again, but from a different angle with a different image, okay? And what we're going to do is we're going to focus on what's going on in this structure here, because notice, even though they're connected and they have these nerves that lead into the brain obviously together, there's a cochlea that is somewhat independent of this whole structure over here, okay? So what's going on in this structure over here? What's going on in the cochlea? So what's going on in the cochlea, remember, is there are fluids that are filling up this cochlea that will sway back and forth or sway forward or whatever, move hair cells as sounds come in, okay? A similar principle is going on in these semicircular canals, except it's a fluid that moves in these canals when you move your head, okay? The fluid here does not care about sounds out in the world. The fluid here does not connect it to the eardrum. The fluid here does not get hammered by the stapes. So the cochlea could be firing on all cylinders, and this fluid won't be moving or doing anything whatsoever, okay? And what's going on is that each of these little semicircular canals is actually involved in different types of head movements, okay? If you twist your head, if you move your head up and down, left, right, so on and so forth, that's what's going to cause the fluid in these different canals to move and to get displaced. So again, fluid here moves from the vibrations from sound. Fluid here moves when you move your head in any of these three ways, okay? Just to try to drive this home, here's another one of these images. Cochlea is down here. You've got three of these semicircular canals. Each one of these ones that are involved in maintaining balance comes from the fact that these fluids will move when you move your head up, down, left, right, or twisting, okay? To give you a sense of what this is like, because this concept might be a little foreign, here's actually a video that I'll just kind of talk us through as we're seeing it. So when you move your head, in this case left and right, basically literally because of gravity kind of shifting the fluid around, you can see that within this particular canal, the fluid will be moving in this particular direction back and forth. The fluid in the other two canals, though, won't really be doing it. However, if we go and we zoom in on this other canal, for example, now we're talking about this one that's on the top, when you rotate your head up and down, now because of the angle of that particular canal, now that fluid is going to be working, okay? So these semicircular canals, they might seem like they're just kind of arbitrarily attached to this in this inner ear formation, but actually they're not. The three of them are at the specific angles because it's like XYZ coordinates of your head twisting in one domain, moving up and down in another domain, moving left and right in another domain, and so on and so forth. And so when I'm talking about a fluid that moves in these canals when you move your head, I mean that literally, quite literally, that as you move around, the fluid will get displaced because of gravity. And just like it was in the cochlea, there are, again, hair cells that respond to the movement of this fluid, okay? And so down at the base of these little semicircular canals, and those little three regions there, that's where these hair cells are that are responding to this fluid. So what I'm going to show you now is just a cross-section of one of these semicircular canals. It doesn't actually really matter which one. So now this is one of these semicircular canals here. And at the base of it is this little thing called the cupula, okay? And that's where the hair cells are, okay? And if you zoom in, if you look at it, you really can see that these little stereocilia look just like the hair cells that we were talking about when we were talking about the cochlea, except now, again, these are not going to get activated by sound. They're only going to get activated by the movements of your head when you do it left, right, up, down, or twisting, and so on and so forth. Okay, same exact principle, fluid dynamics, hair cells, except now this is going to give you a sense of balance. Okay, a natural question that people might wonder is, what about horizontal, vertical movements, okay? Because we did, twisting up, down, left, right, but what about other domains? So it actually turns out that there's also in this little inner ear structure, but again, still separate from the cochlea down here, called the utricle and the saccule. And they're involved in horizontal movements and vertical movements because, if you start moving forward, backward, up, down in that direction, you can still sense it. If ever, you're riding a bike and then all of a sudden you start going forward, you have a sense that your head is moving forward. So the way that this works actually is not because of semicircular canals and fluid dynamics, but because of these little crystals that are called otoliths that sit on top of these structures, okay? That there's this fluid and this blue stuff here with these little crystals on top of it. And what ends up happening is that, say you're on a bike and then you just take off and you move forward, momentum of those crystals will kind of drag back, and that is what causes the hair cells to bend, okay? And then if you slam on the brakes, those hair cells will go, the momentum of these little otoliths will go forward, which will cause the hair cells to bend in the other way, okay? And so what turns out is that it's, again, the same principle in terms of fluid dynamics and hair cells. It's just in this case, it's not because of gravity moving the fluid around. It's because of the pressure from these little crystals and gravity moving them around. Real quick, I just want to remind you that, we were talking about, I think it was in the first of these lectures on audition, about how sound signals have to go through the hindbrain, the midbrain, and the MGN to get to the forebrain. And I talked about it in terms of the midbrain and auditory orientation, but I want to remind you what we talked about way back in week 3 about the hindbrain, because one of the things we said about the pons, which is a part of the hindbrain, is that the pons is this connective bridge, and it is involved in eye movements and balance, okay? And so if you remember way back then, we talked about how the pons is involved in balance. That's because of this sort of system that I'm telling you about, that what's going on, is that the information from your inner ear, not necessarily from your cochlea, that information will get going to the hindbrain, and it's in the hindbrain that information will be provided regarding your balance, okay? So now we're going to take another break, and we're going to move on to smell and taste. Unfortunately, they're rarely studied relative to the other two senses. And so actually what we're going to do is in lecture 4 of today, we'll talk about olfaction, and then lecture 5, we'll talk about gustation, and then that'll be it. It will just be those 5 for this week.